Thank you again for giving me this 25 minutes of presentation. So my name is Erwann Durand. Yes, I belong to CIRAD. CIRAD is a French institute, and I belong also to a joint research unit called Qualisud. So we are based in Montpellier, so in the south of France, very close to the Mediterranean Sea. And today I'm going to give you a talk about the NaDES and how we are using NaDES in our research activity. So put this here. So actually our research activity are mainly focused on two different aspects. The first one is to develop healthy and stable food products, so mostly enriched in polyunsaturated fatty acid, in vitamin and micronutrient because of this essential property of such molecules. And also we are trying to substitute the synthetic antioxidant and preservative by molecule of natural origin. So for this, we are facing a general strategy to understand the lipid oxidation and the oxidation in general in food system. But you may know that more and more consumer and also processor want to introduce polyunsaturated fatty acid, but also vitamin and micronutrient in their formulation because of the consumer preference and also the nutritional and bioactivity property. And do you hear me very well? Yes? You hear me?
Yes, we hear you, no problems.
Okay, okay. Okay, so, but the problem is when you introduce this kind of molecules, the food degradation is a significant increase. So we went to investigate in two different approaches, either in the chemistry, but also in the physics, how the lipid oxidation happen in the food product. So in this context, we would like to better understand by using chemistry how the lipid oxidation occur regarding the structure and regarding the physics of the system that is very, very important to drive the oxidation pathway of the food system. So in this context, we have, let's say, three main topics to address that question. The first one is the understanding. The second one is to search for antioxidant solution. And the third one is the improvement by structural modification, so improvement of antioxidant activity. And I would say that the NaDES may get involved in these two research topic activities. So to look at new antioxidant solution, mostly for regarding the extraction capacity, and also to improve by structural modification and biotransformation the activity of natural antioxidants. So now let's move to in details to NaDES. So I know that the network is quite familiar with this liquid, so I will maybe go quickly. So we don't know whether we are talking about medium, solvent, liquids, but let's say that there are very huge role on the medium, solvent, and liquid, started from the simplest one, that is the water, and passing through the supercritical fluids, the molecular solvent, either biomass derived solvent or fossil-based solvent. And then we move to a multimolecular based solvent, where theoretically the number is expected to form such a solvent is very, very high. And let's say that if we have to find a definition for a multimolecular based solvent, I would say that this is just a mixing of at least two molecules in a different proportion that they may lead to a liquid phase with new property and inherent property in comparison to the starting molecules. So if we have a look on the history, starting from ionic liquid to natural deep eutectic solvents, so we start early in the 20th century, so in 1940, with the discovery of ionic liquid. It's just a mixing of two different compounds, cation and anion. And then we arrived more recently in 2003, thanks to the work of Abbott in Great Britain. We arrived to deep eutectic solvent, where there are basically 4 different types of mixture regarding the composition of starting molecules. But one of them, the type 3, composed of hydrogen bond donor in combination with ammonium salt or phosphonium salt, has significantly caught the attention of one of the researchers, Rob Verput, in 2011, because most of them were composed in choline-based molecules in association with metabolites such as sugar, polyol, and organic acid. And he tried to correlate this discovery with different questions with pending answer regarding the metabolism in the plant and in living organism. And he postulated the assumption that maybe a liquid first phase in liquid living organism may exist and be helpful to promote the metabolism in the plant and in all the living organism in general. So let's say that the definition of NaDES is any combination of metabolites where the mixture is liquid at operating temperature, so metabolite presence in the nature. And then also a suspected role in living organism. For example, so I put 4 different examples of the role in living organism that such a liquid may have: to store, for example, a water-insoluble molecule, to protect plants against harsh environmental conditions, or for the production of nectar, but also to allow biotransformation in living organisms. And I would say that all this assumption was actually supported with experimental experience in the different lab over the past 15 years. So you know that there are different choice of NaDES. I would talk only about hydrophilic NaDES. So let's say that there are more hundreds combination. It depends if you are using different mole ratio for the same combination, but let's say that there are different choice to provide natural deep eutectic solvent liquid phase. But if you have a look on the number of publications in comparison to other such molecular-based solvent, ionic liquid, deep eutectic solvent, you can see that the NaDES, the number of paper, although the border with deep eutectic solvent in literature is sometimes difficult to make, but the number of publications is very, very low. So we are maybe just at the beginning of the story with the NaDES. And I guess this number is expected to increase and maybe reach the same number of ionic liquid in the next few years. So you know that there are different options to prepare and obtain the liquid phase, and we have seen the different options in the first presentation. And all these options are very complementary, and depending on the structure of starting molecule and also the scale of production, one may be advantageous, and the different parameters could also be adjusted. So according to me, the special feature of NaDES, as a liquid phase, is their solubilization and stabilization for different compounds due to their versatile polarity, but also because they are stable over very wide temperature windows, starting far below 0 degrees Celsius to maybe up to 200 degrees Celsius. But the problem sometimes that we are facing with this liquid is the high viscosity, especially with the hydrophilic ones. But this viscosity may be decreased with water addition or using increasing temperature. So here, this is one of the main striking example for me to highlight the solubilization and stabilization of NaDES. So you know that rutin is a polar molecule, but it's almost non-soluble in water because of peace-taking interaction, so intermolecular interaction of rutin that is much higher than the interaction with water. But if you put these molecules in the NaDES, the behavior is significantly different, and the interaction of rutin with the NaDES network is much higher than the interaction, rutin-rutin interaction. So it's very helpful to design a very liquid phase. And regarding the stability, you can see that there are 2 different molecules, curcumin. So we just speak about the curcumin in the last presentation, and also baicalin. And we can see that the stability is much higher in comparison to other solvents. That is why this is the key reason for the success, I guess, in cosmetic, this solubilization property, but also the ability to stabilize or preserve the degradation of active molecules. So in our team, we use NaDES for different applications. So the first one was to enrich the phytochemical and antioxidant profile of natural extracts. So here are different examples of this work on coffee and cocoa, different core product or byproduct or waste. We also use the natural deep eutectic solvent to characterize the phytochemical profile of natural extract. So in comparison, and instead of using traditional method with ethanol and water, we try to implement natural deep eutectic solvent to improve the fingerprint, the molecular fingerprint of extract. And also we use NaDES to formulate and enhance the antioxidant activity. So we worked on the formulation of hydrophilic NaDES using electrospraying method, and especially to formulate beta-carotene in the different nanocapsule. And through this work, we significantly improved the loading capacity of beta-carotene, but also the stability. So now if we move to biotransformation, it means that if NaDES exists in plants, maybe enzyme may be activated. And I would say that the story began in 2008, so just a little bit before the first paper of Rob Vercourt, with the work of Gork and al. In 2008. So it started with hydrolase reaction in DS or NaDES. And our begin starting in 2010 with a model reaction to assess the lipase activity in natural deep eutectic solvent. So that's a very easy method to screen the enzymatic and especially the lipase catalyzed reaction in the liquid phase. And what we observed, so we tested different solvents. And what we observe a very strong effect of the NaDES composition. So now it's obvious, but in 2010, it wasn't very obvious. And we also shown that the side reaction was observed in a natural deep eutectic solvent or eutectic solvent, meaning that we can observe reactivity from the component of NaDES. So the component forming the liquid phase in the NaDES may react in the different reactivity. After that, we try to move to much more complex lipase and enzymatic modification in natural deep eutectic solvent. So we move with the best candidate of medium. And our goal was to biotransform the phenolic compound into what we call phenolipids, because we found that phenolipid may significantly increase the antioxidant activity of parent molecules. So we try to implement biotransformation of phenolic compound in NaDES. And what we observed at the beginning, we observed no direct esterification with phenolic acid. So if we start with phenolic acid instead of methyl ester, we do not observe any biotransformation because of the strong interaction of the acidic group in the network in the medium. And when we move to methyl ester, very low conversion rate was observed in pure natural deep eutectic solvent. But when we put water, so the water content, when we put water content in the NaDES, we significantly increase both the lipase initial activity, but also the final conversion rate. So after that, we went to investigate the effect of water. And as you can see here, so we tested two different mediums. You can see that the conversion rate is significantly increasing upon addition of water, but the hydrolysis reaction remained very low. And the same for the other solvent. So meaning that if we fine-tune the content in water in deep eutectic solvent, you can maximize the phenolipid formation, so the lipase catalyzed reaction. So after that, we conducted a lot of experiments to understand how the water gets associated with the choline chloride and how the water gets associated with choline chloride and the urea, that is the hydrogen bond donor in this liquid phase. And we have seen, so I will go quickly over this result. So, but if you want to go more details, you can check this paper. But what we have shown actually two different zones. The first one corresponding to choline chloride saturation, and the other corresponding to water availability that is increasing after saturation of ammonium salt. And this zone was correlated with the behavior of lipase and especially the stability of lipase, as you can see here. In this zone 1 corresponding to saturation of ammonium salt, we have very low stability of lipase, but when we increase the availability of water toward the lipase especially, we can significantly increase the stability that may be changed with the concentration of urea. So we also tested other reaction and biotransformation in the NaDES, and especially the transesterification of sterol. So in this case, we shown basically the same behavior with a significant effect of water. So meaning that this is very important to check and tune the water content when you are doing biotransformation in the NaDES for several reasons, as you can see here. And we observe also competitive reaction coming mostly from the hydrogen bond donor that is able to react in the such reaction. So let's say that many enzyme now, after almost 15 years, has been tested and shown to be active in presence of NaDES, either as a pure solvent or medium, or as a co-solvent with water or other organic medium. And I would say that the specific enzyme conformation in natural Deep Eutectic solvent is responsible of the different activities, stabilities, and selectivity that we observe in the literature. So I just selected different work that may explain this different result and conclusion. You can see here that the stability is significantly different in the NaDES in comparison to other medium. The same here, and the same also in this work. And regarding also the selectivity, this paper is interesting because it showed that the selectivity for biotransformation of substrate may be changed in the natural Deep Eutectic solvent regarding the composition that we are using. So this approach may be very interesting also. So in conclusion with biotransformation, so there are several drawbacks and several advantages. So I would say that in the advantages for biocatalysis, this is to improve the substrate solubility, to improve the catalytic activity, and especially if you can get specific conformation of enzyme and thermal stability, and also if you want to shift or change the enzyme selectivity. But we have several drawbacks, including the fact that sometimes we need to fine-tune the condition, so the component of the NaDES, but also, as I told you and as I showed you, the water content, that may significantly change both the activity and stability of the lipase. And also the strong interaction in the NaDES that may lower the substrate reactivity, that is very, very important here. And also the competitive reaction that we have to take care, sometimes is underestimated. And also one of the questions about the product and enzyme recovery due to sometimes the very high viscosity of NaDES. So in this context, we went to investigate other strategy to improve the NaDES interest in biotransformation, and especially to go towards sustainable one-pot extraction and biotransformation using NaDES. So here, this is the first example to produce active ingredient like a surfactant, glycolipid. So we try microwave-assisted one-pot lipid extraction and glycolipid production from yeast in sugar alcohol-based media. So in the 3 different steps, the first one was a very fast media preparation with low energy input using microwave-assisted extraction, so choline-based NaDES with sugar molecules. And then we added a yeast to extract the lipid from the yeast. And then with the lipid from the yeast, after acidification and lipase addition, we did a biotransformation of with the lipid extracted from the yeast and the sugar present in the liquid medium to promote the formation of glycolipids. A second example, we use this similar strategy to produce natural extract. So in the third step, again, the phase 1 was to the extraction, the phase 2 biotransformation, and phase 3, we tested the biotransformed NaDES extract to promote the antioxidant activity of rice grain. So we implemented 2 strategies, the strategy 1 and the strategy 2. The strategy 1 with the NaDES that is able to extract and also do a biotransformation, so allow a lipase catalase reaction to modify the structure of antioxidant with lipid that was extracted. And the second strategy was called shiftable NaDES, where we started with very acidic medium. And after very high extraction capacity, we neutralized the system to allow lipase reaction and modification of structural antioxidant to promote the new pool of molecules in terms of antioxidant ability. And this is what we have shown. The biotransformed NaDES extract, after modification of the structure of the molecule as an antioxidant, significantly or may significantly increase the residual antioxidant activity. So now usually we talk with the NaDES and the behavior in single and homogeneous phase. And we know that the behavior of NaDES is very different to other systems. And now the literature is quite unanimous to say that the NaDES in the bulk phase is like it's like non-really homogeneous, and some nanostructure appear in the bulk phase of NaDES that is sometimes responsible of the high extraction capacity of NaDES of such a liquid phase. And the water in both small or large quantity may significantly influence the liquid nanostructure of the NaDES. So if I have to make a sum up of the effect of NaDES in the bulk phase of hydrophilic NaDES, the effect of water, I would say that we are moving from synergetic effect to additive effect sometimes. So if we start with NaDES, we are facing like a superstructure. And if we add other molecules into this network, sometimes we can significantly change the physicochemical property of NaDES that may be helpful for the technological processing. But when we reach very high water content, we are losing the physicochemical property of the NaDES. And this is what we observe, for example, when we try to the solubility effect of molecule, we are losing the solubility effect, the same for stability. We are reaching the same physical and chemical property as the one in water. So the question now is how to get NaDES formulated, hydrophilic NaDES formulated in water-based system. And one of the questions that we started to answer, this is how, if NaDES exists in plant, how NaDES may get interact in the cell that is mostly enriched in the water, how NaDES may get involved, how NaDES may be localized in the cell system to promote their potential in the cell metabolism. So we started for what we know about NaDES, and then what we observe in the cell, and see the connection and how the connection may happen regarding the physicochemical structure of NaDES and what we observe in the cell, that is a medium with different microstructure, but mostly water. So this is take-home message that I would like to forward to ending my presentation. So NaDES, this is metabolites association and liquid phase and the role in living organisms. NaDES is very strong heterogeneity at nanoscale level and very strong effect of component, including water. And NaDES characteristic may be very helpful to implement eco-innovative strategy and new technology, and also could be a new inspiration for understanding and mimicking the nature. So I would like to thank all the partner, all the NaDES partner that who contributed in this work and the result that I presented today. And I would like also to thank everyone. And if you have any question, I'll be very happy to answer. Thank you.